The overall goal of the following experiment is to quantitatively assess the spatial colocalization of target pairs, in this case, examining drug-induced receptor trafficking. This is achieved by treating live cultured cells with drug to induce altered receptor trafficking. As a second step, double immunolabeling of the target receptors and intracellular trafficking compartments allows spatial localization of the target pairs by multi-channel confocal microscopy. Next, quantitative colocalizational analysis is used to measure the tendency of the target receptors and compartments to be found in the same place. The results show changes in receptor trafficking following drug treatment based on the colocalization of receptors and intracellular trafficking compartments.
The main advantage of this technique over existing methods like overlay colocalization is that it provides quantified measures of colocalization, which then allows far more powerful analyses and complex experimental designs.
Though this method can provide insight into receptor trafficking, it can also be applied to changes in the spatial colocalization of just about any two proteins in cultured cells.
To begin this procedure, remove the original growth medium and replace it with medium containing the drug of interest at the chosen concentration. Next, incubate the cells in the original growth conditions for the chosen treatment duration. After incubation, wash the cells gently with 1 ml of washing buffer each time for 3 times. Fix the cells with 300 microliters of 4% paraformaldehyde in 0.1 molar PBS for 10 minutes.